So anyway, let's come to the topic of today. It will be the epigenetic code and their implications in the stem cell biology. And this study is just based on the deep sequence analysis. So in the last century, as we know, that the human genome sequence has been launched with the belief that if we have the whole set of the genomic sequence information, we will be able to understand the development, the disease, and other issues about our human body. But when we get the whole set of the genomic sequence at the earlier of this century, we still find it's very difficult to understand the human biology. So after that, a new effort, so-called the ENCODE project, has been launched with the effort to decipher or to identify the functional elements in the human genome. Such functional elements include the coding regions and the cis regulatory regions, which interact with some regulatory proteins and the other structural elements encoded in the human genome. And also, various genomic approaches are needed to develop. And at that time, I was in the Singapore Institute Genomics Institute, and with the effort to develop some sequence-based genomic approaches to identify the functional elements in the human genome, including such paired and diatech sequencing. We know that about 15 years ago, there is no deep sequence platform, so we still use the single sequence to do this work. Okay, in addition to the genetic regulations, we know that epigenetic regulations also actively involved in the development. Let's give a few minutes to introduce some background of the epigenetics. We know the basic unit of our chromatin is the nucleosome. It consists of the histones, 4 kinds of histones: histone 2A, 2B, H3, and H4, which are around by 146 base pair DNA. The histones are subject to various post-translational modifications such as acetylation, methylation, phosphorylation, ubiquitination, so on and so forth. The different modifications have different effect of gene expressions. So among those different kind of histone modifications, we particularly focus on 2 kind of form of modifications. That's from the H3, K4, and K27 trimethylation. Why we are focused on these 2 guys? Because traditionally it was well recognized that K4 trimethylation is a marker of active gene expression, and the K27 trimethylation is a repressed marker. In other words, this one means open status of gene expression, and this one means the closed off status of gene expression. And from the Drosophila, the fly, to the human being, these mechanisms are highly conserved. So we just try to understand these 2 epigenetic modifications and their implications in the human embryonic stem cell. So we know that the ES cell has 2 fundamental properties. One is the unlimited proliferation, also called the self-renewal, and another feature is the pluripotency, that it has the capacity to develop into any cell type that make up our body. So we try to understand why this kind of cells has these two very specific properties. How to understand their basic properties from their genomic structure or from the epigenetic modifications of their chromatin. So just before our studies, a little bit earlier study from the Whitehead Institute, they used chip-on-chip, just use a microarray to detect just 2.5% of the human genome. They found the ES genome carried a very specific modification pattern which consists of K4 trimethylation and the K27 trimethylation. They termed this modification pattern or this modification code, epigenetic code, as bivalent domain. They find this bivalent domain resolved upon embryonic stem cell differentiated. That means this particular pattern of modification is highly associated with the ES cell stage, but disappeared when the ES cell differentiated. So, based on this observation, we just ask, this result just come from very, very little part of the human genome. It's just 2.5. How about the whole picture of the entire human genome? That means this modification code does this modification code work on the whole genome scale? With these questions, we developed sequence-based analysis, some approaches which enable us to do the expression profiling analysis and the histone modification profiling analysis. Just use the single sequence because at that time the Illumina sequence and other approaches are not available. So just with the single sequencing, we profile the K4 trimethylation and the K27 trimethylation, these two histone modification sites across the human ES cell genome, and identified 30,000 and 100,000 K4 and the K27 modification site. So this is the typical K4 modification pattern. From this pattern, we can see that this histone modification is very accurately located at the promoter region of each gene. And for K27 trimethylation, is totally different pattern. It's very broad rather than this very sharp pattern. And also, we found the so-called bivalent domain, which observed in the Whitehead Institute, consists of K4 and the K27 trimethylation. And this pattern, we know, is highly associated with ES cell undifferentiated status. Then, with this epigenetic data, we just examine how about the status of the genes they market these two epigenetic sites. We found about 67% of the gene are marked by the K4, and 11% of gene are marked K27. The interesting thing is, for those genes marked by K27 trimethylation, nearly 90% are also co-occupied by the K4, and left about 10% without K4 signals. And we ask, are these parts are also marked by K4, but just not detect during the low sequence depths. And we use more sensitive method, quantitative PCR, that means qPCR, to detect K4 signals of those K27 marked genes. And as we expect, all these parts are also marked by K4. So this part can push inside. And that means all K27 marked genes are co-marked by K4. That means all the genes set in the ES cells, they can divide into three parts. The K4 only marked and the K4, K27 co-marked and the genes without these two markers. So next, we try to understand the genes with different kind of histone modifications. How about the expression status? Then we found the K4 only marked genes that are highly expressed. And for K4, K27 co-marked genes, they are relatively expressed at low level. And for genes without these two markers, even much less expression levels. And then last, we try to understand how about this histone modification, different epigenetic code contribute the basic properties of the ES cells. And we do the gene ontology analysis. We found for those K4 only, just genes marked only by K4, they are involved in the nucleoside metabolism, protein synthesis, so on and so forth. Basically, they are related to the cell of the ES cells. And the K4, K27 co-modified genes, they contribute to the development potentials, that means pluripotence of the ES cells. And the genes without these two markers, these two epigenetic codes, they are just involved in the terminal development. So this is the story just almost 10 years ago I did when I was in Singapore Junior Institute. And the next, when I moved back to the China, to the Shanghai, I just started to study the epigenetic regulations and not in another kind of stem cell. That means the female germinal stem cell. We know that for male, the male germinal stem cell is widely accepted for everyone, right? Even in the textbook. But for the female germinal stem cell, it's kind of relatively new, right? Because we know the men even have the ability to give birth, even he is 50 years or even 60 years. But for the woman, it's very difficult to give the birth when the woman is become old. The difference is for men, he has the male germinal stem cell. But for the woman, even in the textbook, it says there's no such kind of germinal stem cell in the woman's body, in the woman's ovary, right? But in the 2009, a research team in the Shanghai, just my collaborator, that professor identified the female germinal stem cell from the ovary in the mouse. This is the first time to identify such kind of stem cell in the mammalian system. Because in the fly, in the C. elegans, it's well established they are female germinal stem cell. But for the mammalian, for almost 80 years, nobody's recognized such stem cell in the female mammalian organism. So this is the first time they identify. Later on, the female germinal stem cell identified in the pig, in the human, in the rat, in other high organisms. So this discovery reshaped our understanding of the germinal stem cell biology. So next, we just want to understand what kind of epigenetic regulations is involved in such female germinal stem cell. And particularly, this kind of stem cell has particular properties, that is the cell renewal that's shared by all the stem cell types, and also the maintenance of the undifferentiated status, and more importantly, why this stem cell has complete unipotence development potential. So we just understand what the genetic and epigenetic regulations defined the female germinal stem cell identity. Again, and at this time we have kind of different so-called epigenetic genomics methods, such as RNA-seq, ChIP-seq, medium or massive-cap-seq, to allow us to profiling the gene expression and histomodifications. So we do find the lineage-specific expression patterns between these two between the ES cell and the embryonic and the female germinal stem cell. And we know that enhancer defined cell identity. So we're just trying to find how the active enhancer in the female germinal stem cell contributed to its identity. Then we just looking for the class 2. That means the female germinal stem cell specific enhancer regions, and do the GO and find they contributed the germinal identity. And also, as I mentioned just now, the K4, K27 bivalent domain play a very important role in the ES cells. But we found in the female germinal stem cell, it doesn't work because it's very low number of bivalent domain genes. Then we try to understand what other epigenetic code. We know for the germ cells, the somatic program has to be repressed in this kind of in the germ cells. So we're looking for epigenetic modification. That means DNA methylation is a well-known repressive marker for gene expression, and we profile the DNA methylation in the female germinal stem cell genome, and we find the methylated genes in female germinal stem cell. These genes are involved in the somatic program. That means in female germinal stem cell, the DNA methylation is used to repress the somatic program and to keep these cells as a germ cell identity. To prove our observation, we knock down the enzyme which responds for DNA methylation, and some somatic genes are upregulated, which shows that in female germinal stem cell, DNA methylation is really important. And also, we compare the DNA methylation pattern from the female germinal stem cell with the male germinal stem cell. And we found that the DNA methylation contributes to maintain the sex identity in the female germinal stem cell, because some female germinal stem cell specific DNA methylation genes are involved in the male development. So this story just tells us that the DNA methylation is a very important epigenetic code in the female germinal stem cell and contribute to maintain the sex identity for this kind of female germinal stem cell. So I think I just stop here.